Good morning, friends. Today we are going to talk about a technique of cytology that is flow cytometry. I am Dr. Vibha Khanna. Now, what is flow cytometry? Etymologically, every word it defines itself. So, flow means motion. Cyto it refers to cell. And when we talk of metry, what we mean is measuring something. So flow cytometry is measuring the properties of the cell while in a fluid stream. This flow cytometry has got multiple applications. Like it can be used for determining various cell functions, the cell complexity or the number of the cell. It can be used for immunophenotyping, for analysis of nucleic acid, that is the DNA and RNA molecules present within the cell. It can be used even for the ploidy analysis, then apoptosis, for sorting and cell isolation, and many other applications of this basic technique. They have been developed, they are being used. So, what is the basic principle of this flow cytometry? The flow cytometer, it performs this analysis by passing thousands of cells per second through laser beam, capturing the light that emerges from each cell after it passes through this beam of light. Now, the data is then gathered and analyzed statistically by the flow cytometer data analyst. This data, it can be used to determine the size, the complexity, the phenotype, etc. of the cell. The range of cell size that can be used for this flow cytometry, it varies from about say zero point 0.2 to 150 microns. That's quite large, big scale. So for this purpose of this multi-parametric analysis of a cell of such wide range, the flow cytometer that is used, it has got certain components. There is the fluidic system, then there is the source of this laser, there are the optics, a combination of mirrors and lenses that direct the light. Then these are the detectors which receive the light from the optics, and the electronic and computer system which converts the signals from the detector to the digital data and perform the necessary analysis. This fluidic system, it provides the sample for the interrogation point and then takes away the waste. So it is the main point which governs the entire process. So for the functioning of this flow cytometer, there are three major parts. First is that the cells, they should pass the laser beam. This is possible because of this fluidic system wherein the cells, they flow in a single file, and at interrogation point the incidence laser beam is scattered by these cells which pass this interrogation point one at a time. Then the optics, it collects the scattered beams, filters them, that is on the basis of the different wavelengths, and this is detected as voltage. Now this voltage is then converted to the digital signals by the electronics, and then they are stored in the computer system for further use and analysis. So what we have seen is that the interrogation point, it is the heart of the system. This is where the laser and the sample, they intersects, and the optics, they collect the resulting scatter, and if we use the fluorophore, even the fluorescence. We'll talk about it in the later slides. Now it is essential that the cells, they pass through this laser beam, that is through this interrogation point, one at a time. This is achieved by hydrodynamic focusing. What we mean by hydrodynamic focusing is that we use the this system, the sheath fluid, to channelize to these cells. Over here, the population of the cell, there are more cells at a single point. They are channelized in the form of a single file. So most flow cytometers, they accomplish this by injecting the sample stream containing the cells into the flowing stream of the sheath fluid or the saline solution. This sheath fluid is also known as the saline solution. The sample fluid and the sheath fluid, these two, they do not mix with each other. Now this introduction of a large volume of into a small volume, here the volume is large, and over here this volume, it reduces because of the diameter of this fluidics system. So this results in focusing or this single filing of the sample cells. This compresses the cell stream into a diameter of one cell, and this is hydrodynamic focusing. So how can cells be made to flow in a single line? For this purpose, saline solution is used. Now from the top, this solution, it goes down from top to bottom, and thus making a directional flow focusing, as it creates a pressure, because according to Bernoulli's principle, when it flows down through this narrower part of the system, it generates pressure. It keeps a constant specific pressure, and under this pressure gradient, the cells, they move down in the form of a single file. Now when the cell, it comes in the path of the laser beam, it scatters this light. This scattering is in all directions, and this scattered light is detected by different detectors, rather different types of detectors. There, you may say that this scattered light can be broadly categorized into two categories. One is the forward scatter or the low angle light scatter. It is the amount of light that moves in the forward direction as the laser, it passes the cell. It is refracted by the cell in the flow channel and then continues along the light path, that is the same direction that the light was originally traveling. The forward scattered light is detected by a sensor in the light path and is typically used to identify the size of the particle. The magnitude or forward scatter, it is roughly proportional to the size of the cell, and this data, it can be quantified to determine the size of the cell. the detector, it converts the intensity of the light into voltage. Now, as the light cell, it passes through the laser, the light is scattered and is collected by the detector. Now, the light which is scattered in other directions, that is not in the forward directions, but in other directions, it comes in contact with the detectors, the side scatter detectors, which are placed at right angle, that is at an angle of 90° from the laser path. Thus, one of these detectors which is in line with the light beam is used to measure the forward scattered, whereas a series of another type of detectors which are placed perpendicular to the stream, they capture the light whose angle is more, that is, and that records the data in the form of side scatter. Now this data that is collected by these detectors, the forward scatter and the side scatter data, it is then analyzed. See, what is happening is, if we see a simple scatter histogram, the forward scatter histogram or the side scatter histogram, it is basically in the form of a pulse. It is plotted of the voltage against the time. And here we are talking of the forward scatter histogram, and we have just talked about that it is used to determine the size of the cell that passes through the laser beam. So, smaller the cell, narrower this path and smaller is this pulse. Larger the cell, this scattering is more and the pulse generated, the voltage is more. Now, if we draw a one-dimensional histogram of this forward scatter, we are talking of this forward scatter. So, if we draw a one-dimensional histogram, that is cell count on one axis and the forward scatter on another, this is basically what we obtain. And if we draw this one-dimensional histogram for the side scatter, these are the different cell counts that we obtain. Now, by itself, these two histograms, the one-dimensional histograms, it is not easy to understand them, because we want to study the cell as a whole. So what we do is we prepare a two-dimensional histogram. Is the forward scatter, that is the size, is plotted against the side scatter, that is the complexity of the cell. And now these different colored peaks, they are produced in the form of this dots. The complexity, more it increases over here, and even the size, the smaller cells, they are towards this left part, and the larger size cells, they are towards the right part of this two-dimensional histogram. Now, one of the parameters that can tell us more about the cell structure is and even its function, it is the fluorescence. Now, if we just revise the concept of fluorescence, it is the term used to describe the excitation of fluorophores. These are the fluorophores. When they absorb the laser, the light, the energy level, it increases. They get excited, and when they come back to the lower energy level, this energy is emitted in the form of fluorescence. And the energy of the emitted light is less, and this excited fluorophore, it again moves down to the ground state. So a common way of studying more of the parameters or cell characteristics is using fluorophores in flow cytometry. It involves using different type of fluorescent molecules which are compatible, which are having different peaks. They are used to label, say, antibodies, and these antibodies then they attach themselves to the cell surface or particular mm, part within the cell, to certain cell organelles or anything, and then they are they flow down in the sample. When they pass through this interrogation point, these fluorophores, they get excited and emit fluorescence. This fluorescence emitted is detected with the help of mirrors, which are again placed at right angle. That is similar to the side scatter detectors. Now, using these filters, we are basically filtering different wavelengths of the fluorophores, that of the emitted light that comes out, that is emitted by the fluorophores when they return back to their ground state. Now this fluorescence data is collected in a similar manner, and then it is analyzed. Now up to different 17 different compatible fluorophores, they can be used parallel in the modern system of flow cytometry. The only thing is that specific band pass filters, they are to be used along with the detectors to detect this fluorescence, so that these different peaks of different fluorophores, they do not interfere with each other, and we get a clear picture of the cells that are under study. So to conclude, we may summarize the entire process of flow cytometry in very simple terms. What happens is that the cell samples, they pass down through the narrow channel in a single file. When they crosses the interrogation point, light, that is laser, it illuminates them, and these cells, they scatter the light. This scattering depends on the various parameters, the size, the shapes, or the complexity. These scattered light rays, they are then detected using basically two types of detectors. The forward scatter detectors, which lies in the path of the light, and the side scatter detectors, which are placed at 90°. These detectors, they convert the voltage signals into the digital signals, and this data acquired is compiled and then integrated to build a comprehensive picture of the sample. So flow cytometry is a unique tool providing scientists with ways to gather statistical data on large number of cells and use that data to correlate multiple parameters within the cell population. Parameters like size, shape, the internal complexity, the number, it can be detected using flow cytometry. Thank you.